Our lochs and rivers are among the cleanest in Europe. The fish found in them are one of Scotland's most important renewable resources. They're priceless assets, and they have to be managed wisely, not only for the fishermen of today, but also for the enjoyment of future generations. Effective conservation needs a proper understanding of the factors which control fish populations and their ability to survive fishing pressure and environmental change. That need is being met by the scientists of the Freshwater Fisheries Laboratory at Pitlochry. The main laboratory is at Fascally, with specialist units at Montrose, Ballater, Armand and Aberdeen. And their aim? To provide the Scottish office with the information it needs to safeguard both the freshwater fish populations themselves and the rivers and lochs in which they live. It's a major undertaking, involving a complicated network of investigations, the results of which are vital to everyone who uses the resources, from anglers to netsmen and fish farmers. The scientist's job is not made easier by the fact that the life cycle of fish, like the salmon, is so complex. In late autumn and winter, eggs are buried in the gravel. These underwater nests are known as redds. Investigations begin even at this early stage.
Each salmon redd contains about 5,000 eggs, and a technique of redd excavation allows us to remove a small sample of eggs from each redd. In this case, we take about 20 to 50 eggs from each redd. By analysing these embryos, it's possible to tell more about the parentage of these eggs than was previously thought possible. In addition to the adult male fertilising the salmon eggs, precociously mature par additionally fertilise eggs, and as many as 5 or 6 different fathers can be found in the same batch of eggs. It's possible also in the future to DNA fingerprint each of these individual eggs, and this will allow us to work out in much more detail than was ever possible before the fate of each family's offspring during their time in fresh water.
By spring, the eyes of the embryo begin to show in the developing egg. And eventually, usually in April, out of the eggs come young alvins, and the second stage of the cycle begins. First of all, the alvins absorb their yolk sacs, then move up out of the gravel into flowing water, where they'll spend between 1 and 5 years as par. During this stage, a technique called electrofishing is used to sample the young fish in the burns. It lets the scientists see how many fish are there and what kind of habitat they prefer at different stages of the development.
We're on the Tay system in Perthshire. This is a typical spawning stream for salmon and sea trout. It's very important to know the numbers of young fish on which future runs of salmon and sea trout will depend. And in a small stream like this can be easily done by electrofishing.
Some of the larger juveniles can also be marked. For instance, a tiny metal microtag can be inserted under anaesthetic into the nose of the fish. This is one of the more recent tagging methods used by the Pitlochry scientists to follow the fate of the fish. The next stage of the cycle begins when the juveniles start to swim downstream. For some young salmon, this migration starts in the autumn, when they leave their home burn for the main river. In spring, they're joined by others to form the smolt run. Near the mouth of the North Esk, some of the smolts are caught in a specially built trap operated by the Freshwater Fisheries Laboratory at Lade. Here, they're tagged and released into the main river, a proportion of them above the entrance to the Lade. Using this system, the scientists can estimate the total number of smolts leaving the North Esk from the proportion of previously tagged smolts in the sample caught in the Lade. Over the years, the numbers of smolts leaving the North Esk have varied, but there's no evidence of any strong, long-term trends up or down. Some of the tagged fish, by this time much larger, are recaptured in fisheries, including those at West Greenland and Faroe, where fish from Scotland mingle with those from many other countries. By studying the pattern of tag returns, the scientists can account for the losses suffered by the salmon as they pass through a succession of fisheries, beginning on the high seas, continuing along our coasts, and ending with the angler's lure in the river. The tag results are combined with the catch statistics of the fisheries and an independent electronic count of the total North Esk run. The scientists can then estimate the levels at which the stock is fished and find out whether enough adults have survived to make full use of the river's spawning and nursery areas. Collecting the catch statistics is one of the laboratory's most important and most complex tasks. Forms are sent out each year to fishery proprietors all over Scotland, and national statistics are compiled on the basis of their reports. The electronic counting of fish is a newer art. It's made possible by the fact that the electrical resistance of a salmon's body is less than that of fresh water. This is a recently perfected fish counter at Logie. Thanks to its advanced electronics, it can compensate automatically for changes in the conductivity of the river water and so recognize when fish are passing as opposed to other objects. Following the success of the Logie fish counting weir, similar weirs are being installed not only in Scotland but also in Spain and North America. The scientists can summarize the data they collect from their tagging, from the fisheries and from the fish counter in the form of a simple life table, which records the fate of a generation of salmon. By combining successive life tables, they can see whether the stocks are limited by the number of spawners or the amount of food and space available for the young fish. The results have shown that in well-stocked systems, such as here at the Shelligan Burn, there are enough young salmon to make full use of the available habitat. The laboratory is often asked whether salmon runs can be increased by stocking. To give an answer, researchers first conduct a survey to see whether there is spare nursery habitat for the extra fish. If there isn't, then stocking isn't beneficial. Instead, improvements will depend on better habitat management, especially of bank sides. Alternatively, changes in the level of fishing may be necessary. Stocking comes into its own when the numbers of adult salmon are too low to stock a river or tributary naturally. For example, when there's some obstruction. In instances like this, man can give nature a helping hand. Broodstock are gathered at sites as close as possible to the part of the river that needs to be stocked. It's important to select broodstock carefully, because there is increasing genetic evidence that over many generations, salmon have become adapted to their home rivers.
The Atlantic salmon in Scotland is an assemblage of more or less reproductively distinct populations, and each population has a genetic constitution which is adjusted by natural selection towards maximizing survival and reproductive success in that population's river. Differences among salmon populations can be studied by direct analysis of cellular DNA, the molecule where the genetic information is encoded. It's the existence of adaptive genetic differences among populations which makes the choice of the source of brood fish for stocking a crucial concern.
Eggs are collected and incubated. Decisions then have to be made as to when the developing fish should be planted out. For some, it'll be at this early stage, as fry, just as the yolk sac is disappearing. Others will be introduced into the population as bigger juveniles and smolts. We've seen that the freshwater environment sets strict limits on the numbers of smolts migrating to sea. For this reason, environmental investigations are as important to the Pitlochry scientists as studies on the fish themselves. One of the best-known environmental problems, studied recently, is acid rain, or more correctly, surface water acidification. A study of the remains of microscopic plants in the sediments at the bottom of many of Scotland's lochs has shown evidence of increasing acidity since the beginning of the Industrial Revolution. Over the past 60 years, this change has speeded up, and it's coincided with the reduction and disappearance of trout populations in several lochs in southwest Scotland. When livestock have access to riverbanks, the vegetation is trampled underfoot, and root systems which help to hold the banks in place break down, and so much soil and mud may be washed into the river that eggs and alvins are smothered. The clear felling of forests can have much the same effect if care is not taken to protect watercourses. Here, the accumulation of brash may even obstruct the movement of adult fish at spawning time. The most important single contributor to freshwater acidification is airborne pollution, which comes mainly from sulfur and nitrogen in industrial waste gases. In Scotland, the most acid pollution is found in the east of the country, but the soils there are high in lime, and this neutralizes the acidity before it reaches the rivers. In parts of central and western Scotland, however, where slow weathering rocks like granite and schist are covered by shallow peaty soil, there's not much lime available to counteract the incoming acidity, and its effects are much more serious. In conditions like these, conifer plantations may increase the acidity of catchments. They collect acid mists and gases very efficiently, and the acid is then washed down through the soil and eventually into flowing water. When trees are planted as densely as this, they take up lots of water, so that stream levels are lowered and, in summer, some streams even dry out. By contrast, the rapid runoff from the forest drains can lead to flash flooding and the clogging of spawning gravels by silt. Trees which are planted right up to the edge of burns also cut down the light getting through to the water. Heavy shade inhibits the growth of vegetation, which is an essential part of the freshwater food chain. Paradoxically, though, planting broadleaf trees along riverbanks can improve conditions for fish. This is because, unlike conifer needles, their leaves decompose readily and help in the production of fish food organisms. Deciduous trees are also a good source of terrestrial insects, some of which fall into the water and are eaten by fish. Fish farming is a much younger industry than forestry, and it's ironic that its requirement for clean water can also lead to adverse effects on the freshwater environment. For example, when fish tanks are cleaned out, suspended particles can silt up spawning grounds. And there are other, more subtle effects. Along with phosphates, which seep into the water from agricultural activity on the land around lochs, more nutrients are introduced by the processes of fish farming. These boost plant life and increase the numbers of some food organisms with consequent effects on fish. All these factors have to be investigated. One of the ways of doing that is to reproduce the different sorts of environment in laboratory conditions. Here at Almond Bank, things like light, temperature, salinity and so on can be carefully controlled in tanks, flumes and stream channels to simulate different habitats. The behavior patterns of the fish are then recorded on closed-circuit television for detailed analysis. At the same time, larger-scale experiments are done in the rivers themselves. For instance, traps have been installed on 2 small tributaries of the Aberdeenshire Dee. Parr and smolts leaving the stream are caught and tagged. Adults returning to spawn are also caught and checked for tags before being allowed to continue upstream. This enables the scientists not only to relate the numbers of adult offspring to the strength of their parental generation, but also to estimate straying. It is because relatively few fish stray that inherited differences between the populations of salmon and trout occupying different river systems tend to persist.
The young salmon caught in the trap behind me are on their way down to the sea. About 3,000 or 4,000 of them make this migration every year. All of them leaving the stream are micro-tagged, and when they return as adults to the stream in a couple of years' time, the tags will reveal whether they've homed accurately. Early indications from micro-tagging work is that homing is more precise than we even imagined. Not only do fish make it back to their native river, they home precisely to small tributaries such as this. As many as 90% of the females returning to this stream bear tags, and as many as 40% of the males. This suggests that even a small stream like this has its own little population of salmon, which return repeatedly, generation after generation, to lay their eggs here in this stream.
Although assessing the fish populations is a crucial element in the research programme, equally important is the sampling of the other life forms which share the underwater environment of the fish and contribute to their food supply. This simple technique for sampling aquatic invertebrates is called, logically enough, kick sampling. A hand net is held downstream and the operator simply kicks the bed of the stream. This releases organisms like mayfly nymphs and worms from the stones, which then drift down into the net and are collected for analysis. Hess sampling is used to estimate the density of insects present. A cylinder is placed on the stream bed. The bed is stirred up as before. This time, however, because the cylinder has a known diameter, it's possible to count the number and type of animals in a measured area. Vertical haul nets are used to sample zooplankton. They're lowered to a given depth, then pulled back up. This time, it's a column of water that's being sampled. The microscopic creatures collect in the filter. The same principle operates in this Jenkins sampler, but this time it's the bottom of the loch that's being sampled. The cylinder is pushed down into the sediment, the top and bottom traps close and bring up a measured amount from the floor of the loch. And of course, the results of all these sampling techniques have to be processed back at the laboratory. Chemical, physical, biological and statistical analyses are used to build up models of the different environments and the various underwater populations which inhabit them. Samples are sorted and the microscopic invertebrates are separated from plants so that the laborious process of identification and counting can begin. And all this information is transferred to computers for statistical analysis and presentation in the form of charts, graphs and a data bank which is essential for the conservation of Scotland's freshwater habitats. So far we've concentrated on salmon, but this is by no means the only fish of interest to the Pitlochry scientists. Their remit covers all of Scotland's freshwater fishery resources, including eels, pike and Arctic char. One of the most widespread members of the Scottish fish fauna is the trout. It has a very flexible life history, some fish spending all of their lives in a single tributary and others making more or less extensive migrations into rivers, lochs, estuaries and the sea.
Trout have been an important part of our research programme from the very earliest days. We were set up, indeed, as the Brown Trout Research Laboratory just after the war. They have a very flexible life history and exist in a number of different races, some of which seem to indicate the effects of the ice movements at the end of the last ice age. We study them from big sea trout in the northwest and the fast-growing trout of our big east coast sea trout rivers to tiny little populations right up in the high hills. We study them for their own sake and also because they are useful animals as models for our population ecology experiments.
Trout which migrate to sea as sea trout have special economic importance. There's been a decline in sea trout catches in some rivers. Pitlochry scientists are trying to find out if there's been a real drop in the total numbers of trout in these rivers or merely a reduction in the proportion migrating to sea. The problem is very complex and there are many factors, including climatic change, which could be affecting the size and migratory behavior of the stocks. Once again, research is fundamental to understanding what's going on. These fishermen are in fact Pitlochry researchers. One of the most cost-effective ways of sampling sea trout is by rod and line. From each site, the researchers want about 20 fish. They need to know what condition the fish are in, whether they're fat enough, how they're growing, and how many sea lice they have on them. So the fish have to be caught and kept alive until they're ready for disease testing. They mustn't be handled, because this will wipe away the sea lice and make it more difficult for the scientists to interpret any disease symptoms.
Well done, Steve.
Thanks, Andy.
But you're supposed to be catching finnick, not... Oh, what do you think it is?
I think it's a farm fish.
Yeah, it's a farmed salmon. It's an escapee salmon from one of the fish farms. Can you hold that?
Yeah.
I'll point out some of the features. You probably know them already, but just to reinforce the point. The distorted fins are an obvious feature of the farmed fish. Quite often these escapee fish have got rounded tails. Instead of the sharpness of the wild salmon or grilse, these have got rounded fins. And you see also the dorsal fin is eroded badly. And that's not a clean, sharp fin the way that it should be with a wild fish. These fins are ragged as well. See, same sorts of things. Every one's the same fins.
Quite a mess.
And look at the numbers of spots. See, that's not normal with a wild salmon or a grilse. Sometimes a previous spawner has more spots than a maiden fish, but these farm fish are characteristically spotty. And then on this side, there's a wound. Well, that might not be because it's a farm fish. That might be because it's been stabbed by a heron. It's just difficult to say, or a gull. But anyway, well done. But make sure you catch a finnick the next time.
I'll try. I can't promise anything.
It's a reminder, though, of the fact that all the research we've been talking about is concerned with practical, hands-on matters. The idea is not only to propose elegant theories, but to produce first-hand evidence about actual habitats and conditions and to make recommendations that will help to safeguard stocks. Adult salmon run the gauntlet of fishermen's nets in seas and rivers, of anglers' lures, hydroelectric schemes, industrial pollution, and the results of afforestation, agriculture, and fish farming. The Scottish Office, district salmon fishery boards, pollution control agencies and individual fishermen, anglers and fish farmers all benefit directly from the findings of the Freshwater Fisheries Laboratory. Ecological balances continue to shift, and environmental changes present new threats to the fragile habitats on which aquatic life depend. The importance of combining research and management to conserve Scotland's freshwater fisheries cannot be overstressed. Like other natural resources, they're only renewable if we take care of them. And that care has to be based on a sound knowledge and understanding of all their characteristics. This is exactly what the researchers at Pitlochry are working to provide.